So in the previous video we were discussing about the important classification of apoptotic proteins. Now we'll be discussing how these proteins will act. So the DNA damage in DNA or endoplasmic reticulum stress is the sign for apoptosis because of accumulation of misfolded proteins. So BH3 only proteins, as we have seen before, I think we have seen somewhere. Yes, regulated apoptosis initiator proteins will have only BH20 proteins. So these proteins are upregulated through increased transcription. Okay, and hence they will directly activate this BAX and BAK. Okay, they will directly activate two critical proteolytic family BAX and BAK, which form oligomers. This will get inserted. This will get inserted into the mitochondrial membrane and allow proteins from the inner mitochondrial membrane to leak out into the cytoplasm. Okay, so BX and BAK, what they will do? They will allow the proteins from inner mitochondrial membrane to leak out in the cytoplasm. BH10 proteins will also bind to and block the function of BCL2 and BCLX1. So what are BCL2 and BCLX1? These are anti-apoptotic bodies, BCL2 and BCLX1. So it will block the function, and at the same time the synthesis of BCL2 and BCLXL may decline. Okay, so the net result is that BAX and BAK activation will result in loss of protective functions of anti-apoptotic proteins, and they get released in cytoplasm, such as cytochrome C. Apoptotic proteins will get released in cytoplasm and activate the caspase cascade. So cytochrome C, what they will do is they will bind to a protein called APAF, one of the apoptosis activating factor 1, which will form a multimeric structure called apoptosome. Formation of apoptosome takes place. Apoptosome is nothing but when cytochrome C binds with APAF, one is apoptosis activating factor 1. Now with this will bind to caspase 9, which is a critical initiator caspase of the mitochondrial pathway and promotes its autolytic cleavage. So binding of apoptosome with to caspase 9 will promote autocatalytic cleavage. So it will generate catalytically active forms of the enzyme. This active caspase 9 will trigger a cascade of caspase activation by cleaving and activates other procaspases, so which will mediate the execution phase of apoptosis. That's it. First step is formation of apoptosome, next the binding of apoptosome to caspase 9, and then caspase 9 becomes active. It will trigger and activate other procaspases, such as caspase 3, and which will elicitate the execution phase of apoptosis. So other mitochondrial proteins with names like SMAC and DILBO enter the cytoplasm, where they bind to neutralize cytoplasmic proteins and then function as physiologic inhibitors of apoptosis. Okay, so IAP. So IAP's inhibitor of apoptosis, the function is to block inappropriate activation of caspases, including caspases like caspase 3, and keep the cells alive. So IAP inhibition permits initiation. This should be inhibited for the activation of caspase cascade to takes place. Next, let's see about extrinsic pathway. In extrinsic pathway, it is initiated by engagement of plasma membrane death receptors. So death receptors are members of family of tumor necrosis factor receptor family. These death receptors belong to tumor necrosis factor receptor family, and they contain a cytoplasmic domain involved in protein-protein interaction. So death domain is essential for delivering apoptotic signals. Okay, so some tumor necrosis factor do not carry cytoplasmic death domains, so their function will really activate inflammatory cascades. So this is given in detail, but what we want to remember is plasma membrane death receptors engagement. There is activation of plasma membrane bound death receptors is the main role in extrinsic apoptosis. That you have to remember. So the best known death receptors are type 1 TNF receptor, TNFR1, tumor necrosis factor 1, and the related protein called FASCD95. Okay, so it's an example of death receptor. The mechanism of apoptosis induced by these death receptors is well illustrated by FAS. Okay, so the ligand for FAS is called FAS ligand, FASL. FASL is expressed on T cells that recognize self-antigen and function is to eliminate self-reactive. Okay, so FASL, what this is a ligand. Okay, it is expressed on T lymphocytes. Very important. T cells. What will these T cells do? It will recognize self-antigens and functions to eliminate self-reactive lymphocytes. That also express the receptor FAS upon recognition of self-antigen. Okay, so that is very important. And when FAS ligand binds to FAS, the 3 or more molecules of FAS are brought together. That is very important. 3 or more molecules of FAS brought together, and the cytoplasmic death domain forms a binding site for an adapter protein called FADD, FAS associated death domain. This is also important, FADD. Okay, and when this FAS molecules attached to the FADD binds inactive caspase 8, bringing together multiple caspase molecules and lead to autocatalytic cleavage and generation of active caspase 8. Extrinsic apoptosis pathway can be inhibited by a protein called FLIP, which binds to procaspase 8. It will bind to procaspase 8, block the FADD binding and cannot activate the caspase. See here, extrinsic and intrinsic pathways of apoptosis are initiated in fundamentally different ways by distinct molecules, but there may be interconnections between these two pathways. See, in hepatocytes and pancreatic beta cells, caspase 8 produced by FAS signaling cleaves and activates the BH2 only protein BID, which then feeds into mitochondrial pathway. Okay, so there is a link between extrinsic and intrinsic pathways. So example, caspase 8 produced by FAS signaling, it will cleave and activates BH3 only protein. This we have seen the BH3 only protein in intrinsic pathway also. So where it comes in, BH3 only protein comes in regulated apoptosis initiators in intrinsic pathways. Okay, so there is a connection. Next, in execution phase of apoptosis, both intrinsic and extrinsic pathways will join together to activate a caspase cascade that will mediate the final phase of apoptosis. So the intrinsic mitochondrial pathway, we know that it activates initiator caspase 9, whereas extrinsic death receptor pathway activates caspase 8 and caspase 10 is very important. So we should remember the number properly. Caspase 8 and caspase 10 are activated by extrinsic death receptors. The intrinsic mitochondrial pathway will activate initiator caspase 9. That is very important. So the active forms will trigger the rapid sequential activation of executioner caspases. So caspase 3 and caspase 6 or executioner caspases. Okay, that is also very important. And once activated, these caspases cleave an inhibitor of DNase, making the DNase enzymatically active. So cleavage of enzyme that inhibits DNase will lead to activation of DNase enzymatically and allow DNA degradation to commence, and caspases will also proteolyze structural components of nuclear matrix and promote fragmentation of nuclei that we have seen before. Okay, and other steps in apoptosis are less well defined, and removal of dead cells we will see. So the formation of apoptotic bodies will break the cell into small fragments, and which is consumed by phagocytes. So the changes takes place in the cell membrane. Okay, these changes in the membranes will activate the phagocytosis so that they are cleared before they lose before the release of cellular contents and before they lose membrane integrity. Okay, phosphatidylserine is present on inner aspect of plasma membrane in case of healthy cells. Okay, but in apoptotic cell, this phospholipid flips on the outer layer of membrane, and this phosphatidylserine which is usually present inside is expressed outside, and hence it is recognized by several macrophage receptors. Okay, so that is, and also cells that are dying by apoptosis will secrete soluble factors which will attract phagocytes and macrophages themselves, and leading to an engulfment takes place. And apoptotic bodies may also become coated with natural antibodies and proteins of complement system, which are recognized by phagocytes. Next, let us see what is efferocytosis. See here, the numerous ligands which are induced in apoptotic cells will serve eat me signals and are recognized by the receptors on the phagocytes. Receptors on phagocytes will uh, get eat me signal from the numerous ligands bound to apoptotic cells. Okay, so the process of apoptotic cell phagocytosis is called as efferocytosis. This is very important. It is so efficient that when dead cells disappear often within minutes without leaving a trace. So that's it. So apoptosis is over. So now let us see about other mechanisms of cell death and autophagy in the next video. Thank you.